I was slow to get it reset. So I'll try and pick up where we left off. We were talking about sampling methods, and I think where we finished off was talking about multi-habitat kick sampling. Okay, where all habitats are sampled in proportion to their occurrence in your sample reach. So for your report, it was a 2-minute multi-habitat kick sample. And again, that time was apportioned according to the different habitats in the stretch. Okay. Now, Sorber sampling is great because it's quantitative sampling. But it doesn't always work everywhere, particularly if you're dealing with very large substrates. So if there's boulders, you simply can't get that device down on the bed of the river securely enough. So it's essentially like a pond net with a quadrat in front of it. And so what you do is you place it on this the bed of the stream. Let's go back. There it is. So it's placed on the bed of the stream, and then you remove everything from within the quadrat. So you're sampling a defined area. Most of the Sorber samplers cover an area of 1/16 of a square meter. There are bigger ones and smaller ones, and everything then is removed from within that area, and therefore you can express the number of animals found in that area. You can multiply it up to express the numbers per meter squared. There is another device, and I thought I had a photograph of it there, which samples not a square quadrat, but a circular area, and that's known as a HESS, H-E-S-S sampler. And it's a much smaller area. It's only about 1/4 of the size of your frame there. But it allows you to get in between boulders. So that's the reason that it has been developed. But most of us use the Sorber sampler where it's appropriate. The one in that photograph there is it's a much larger area one, and that one we made larger to sample for juvenile crayfish. I'm going to talk about sampling for crayfish in just a moment. Okay, so generally what you do in the field is you collect your sample in your pond net or the net of your Sorber sampler, and then you transfer it to some sort of container. I mean, I generally use these heavy-duty plastic bags. And the sampling, while it might be 2 minutes of kicking, it's always accompanied by sorting or searching for animals that are attached to rocks within your sampling area. For example, your caddisflies, a lot of them stick very firmly to the rocks. So you have to do generally a 1-minute hand search. Now, when it comes to the lentic habitats or the lakes and ponds, it's a different approach because you have shallow water on the margins or the littoral zone, and then you have the deeper water in the profundal zone. So in the shallow margins, we generally use our pond net, and it's again, most people would deploy maybe a 2-minute kick sampling approach. But it's a little bit more difficult because you have to keep moving a lot more because you don't have the water flowing through your net. So you have to move the net to generate the flow through your net and therefore capture any animals that you dislodge. For deeper water, we generally use what is called a grab sampler, G-R-A-B, and there's many different designs. One of the most common is known as an Ekman grab sampler, and it's like a big Jaws. And again, when you're able to come in on campus, I'll show you this and how it works. So the jaws go down in the open position, and then you drop. So if you're in a boat, for example, you drop this grab sampler to the bottom of the lake or the pond, and it has a rope attached to it, and then you let go what is called this messenger. So it's a heavy weight, and as soon as it hits that structure there, it causes the jaws to snap close. Now that type of sampler is great if the bottom of your lake or pond is soft sediment and it doesn't have boulders, and it doesn't have vegetation. If it has any of these things, it prevents the jaws from closing. Okay. Another type of sampler is just called a passive sampler or passive sampling. These are drift nets, and they're just suspended in the water to catch anything that is floating past in the water. So all of the invertebrates, they don't move very fast, they do drift at certain times of the day, and they can be caught in nets. But what's even more important to catch are what we call the exuvia, the skins of some of the emerged or the molting invertebrates, and you can tell the species very easily from those skins. And what's useful about the skins is that it doesn't have all the tissue in it, so it's easy to see through structures. So some people deploy passive sampling, particularly for work on chironomid larvae, which are difficult to identify, or chironomids, which are difficult to identify as larvae. But if you can get the pupal skins, you can easily identify them to species level. Okay, now I'm missing a slide there. What I will do is, on the next day, I'll show you a few more slides. If you're working in lake systems and you're dealing with trying to catch invertebrates that are, say, faster moving or swimming, for example, some of the great diving beetles, they're very hard to catch by kick sampling. So we put in instead what we call activity traps, and I'll show you the next day some photographs of an activity trap. They're basically a Coke bottle with the top cut off and inverted, and the animals swim into it and they can't get out of it. And so if you combine that with your kick sampling, then you get a sort of a better measure of the whole diversity of invertebrates present at the site. But one special invertebrate is the crayfish, and in Ireland we only have one species, hopefully and thankfully, the white-clawed crayfish. It's a protected species right across Europe. It's really under threat in countries outside of Europe, outside of Ireland, mainly from pollution, competition with other invasive species. In Ireland, it has problems with disease, and some populations have been wiped out. I suppose you need to be aware of it, particularly if you're doing any works or investigations where this species is present. Then you often have to take account of its presence and give some measure of its say, relative abundances. So it's a species that lives in water, but it also depends on river banks as a habitat. And some, particularly the adults, will burrow into the river banks. So they live, and you can see these crayfish burrows. So they live, the burrows are damp enough for them to be able to survive. And so they'd rest up in those burrows during the daytime and then get back into the river at night time to forage. So how do we collect and sample crayfish? We use, for the adults, we use these traps, which are very like lobster pots. They're known as, believe it or not, trappy traps, T-R-A-P-P-Y traps. And so they have these sort of two pieces that join together like that. And generally what we do is we line them with a mesh just to prevent, in case we capture some of the juveniles, to prevent them from escaping. So what these guys are doing here is that they're actually baiting the traps. So we put in things like liver or cat food in, and that attracts the crayfish in. And then we bury them in the river, leaving them for, generally 24 to 48 hours. And then we come back and we examine what is contained in the traps. So we measure the length of the crayfish, and we don't measure the entire length. We measure what is called the cephalothorax, so that's the head and the thorax. So it's the area. I don't know whether, can you see my cursor? Yeah.
Yep.
Yeah. So we measure from there to the end of the thorax. That's the measurement, because it's very difficult to stretch the animal out and measure it quite quickly. So we generally measure the length of the cephalothorax, and we take a weight of the animal and check then for diseases or not. Okay. So juveniles are a little bit more tricky because they bury themselves down in the sediment and they're not as readily attracted to the trappy traps as the adults. So we have used instead that very large sorber sampler that I mentioned. So again, it's a case of collecting all the material within the quadrat into your net, then taking it to the river bank, spreading out the substrate material. Because you're dealing with a living sample, the little juveniles just pop up and you can count the numbers that are present. So it's a relatively quick way of counting juveniles. So generally what we do is we would take those sorber samplers on a transect right across the river. You can see the string across the river. If you're using the trappy traps, you can't express absolute abundance, but what you're expressing instead is catch per unit effort. So it'll be the numbers per trap per 24 hours. And so that's a measure of relative abundance. It's semi-quantitative, and it allows you to make comparisons between sites. Okay. Just want to show you that photograph there. Obviously there is a habitat underneath the riverbed. That's known as the hyporheic habitat. And it's an area of saturated sediments, so the space between the sediments. It's a habitat that lies between the surface water and the groundwater. It's an area of exchange between surface and groundwater, with water moving in both directions. And it's a very important habitat for the juvenile stages of some of the invertebrates. It's also a habitat where the invertebrates can escape during periods of flooding, for example. And this is one of the reasons I always say to students, never do kick sampling immediately after a flood event. Leave it for 2 or 3 days, because the invertebrates have gone down into the hyporheic zone and they'll take time to come back up when the water levels drop. Okay, so antecedent weather conditions are so important to note as well. But, there's a whole community of animals that are unique to that hyporheic zone. And this is how they're collected. This is known as a Bois Rouge pump. So it's essentially a pump where we're pumping water from that zone and sieving it. And there's tiny little invertebrates that live in that there. Okay, that's a piece of work that hasn't been done too many times in Ireland. That's a former student of mine, Samuel Kibishi, who did his PhD on that zone. Unfortunately, Samuel, 2 months after completing his PhD, died from cancer. So anyway, we remember him in these slides every time. Okay. Substrates, if we get a chance to go into the field, hopefully we'll be able to define these substrates. It's just something that has to be recorded in your field sheets. And if you remember what you did on that. So it'll be the cover of the different substrates. So the cobbles are the hand to half hand size. Gravel then is finger to half finger size. Sand we know, and the boulders are those larger rocks. So those are the main substrate categories that we have to record. What you didn't get an opportunity to do, and hopefully we will do it, is do a little bit of sorting and just see how that process is run. So it's important that at least we go through the method with you. So you have this big bag of sediment that you've collected from the river, brought back to the laboratory. You have to go through it. And you have to go through it in small amounts. That is, I suppose, where you can see between the pieces of cobbles and gravel and so on, and other debris that's in there. So we always sort a small amount of a sample on the tray, and we work on a white tray, and the white tray is always illuminated. Okay, and then what you've done then is you've used that information to come up with a rating of water quality. Okay, does anybody have any questions on that? That's sort of general guidance, and hopefully we will get out to actually do some of that work in the field. Okay, so that should be enough information then for you to write up the methodology for your report. Okay, no questions. Okay. Now, I put up a file on multiple stressors. It has audio on it. Some of those slides I had given you before by way of introduction, but now you have the complete package. I just want to go through a few of those slides today, not all of them, just to clarify a few aspects. So I'm going to start with that slide. Hold on, way back. Okay, you remember the DIPSER framework that we outlined? So your drivers, pressures, state, impact, and so on. So just to remind you, and we just get their language correct, the drivers are the land use activities that generate pressures, for example, diffuse and point pollution, flow regulation, climate change, invasive species, and so on. And each of those pressures may deliver a whole range of stressors, individual stressors to river and lake systems. So if you take diffuse and point source pollution, that can result in inputs of nitrogen, even different forms of nitrogen. Each has its own stressor effects, phosphorus, sediment, pesticides, heavy metals, and so on. So long story short is that most of these pressures deliver multiple, what we call multiple stressor cocktails. And obviously, you know, when you're addressing water quality problems, you need to identify what is causing the impact so that you can remove that stressor. So that's why we deal with it. And, you know, stressors have been defined in various ways, and I've just put those two definitions are the ones that I would most commonly use. So it's a variable that, as a result of human activity, exceeds its normal range of variation and affects individual taxa, community composition and structure, ecosystem functioning, and all that relative to a reference condition. Okay, we're clear enough on that. I'll give you links then to some of the papers. I mean, there are two topics that are going to be part of your final end of trimester assessment, be it essay or not. There will be an essay there, whether that essay is online or whether we have an opportunity to do some in-person exams here, or on-campus exams is what I should say. And I will either way be giving you the essay titles in advance. And one of those essay titles will certainly concentrate on sediment and multiple stressors. So multiple stressors is going to be a topic that's covered, in fact, probably by both subjects. So it is important that you do the reading that comes with this and that you get a good sense of what we're dealing with. Now, if you take a situation where we have two stressors in a water body, so how do these stressors react, or how can they potentially interact? So there's a number of ways. So one stressor may first of all dominate. So it has the strongest notable ecological effects. Sediment would be one of those generally. Or the two stressors may act independently. So in other words, the overall effect is equal to the sum of the single stressors. So that's what we call an additive effect. Or there may be interactions. So this is where where one stressor affects the expression of another and vice versa. So those interactions may be, first of all, synergistic. So you end up with a larger cumulative effect relative to the individual stressor effects. So you end up with an effect that is bigger than the additive effect. And I'll show you some of those in a moment. Oh, this is timed. Or it may be antagonistic, therefore the ultimate effect is less than that which you get from the simple sum of the two. And it may be either positive or negative. And again, I'll talk about that in a moment. Or we can get what are called reversals. So the effect of one stressor is reversed by another. And that leads to what are been described in the literature as ecological surprises, totally unexpected outcomes. So, now I'm just going to go to this slide here because it summarizes those effects. And I just want to make sure that you understand them. So the CT, so let's think about this top graph here as number of stoneflies. And we're dealing possibly with organic pollution. So in the control, we have 30 different species. We'll call them species of stonefly. When we add, if we had a stressor A on that environment, it would reduce the number of stoneflies to 25. And stressor B would reduce them to 15. If those two stressors occurred together, and if their effect was additive, then we would be reduced to just 10 species. Now, do you know where that 10 came from? Please say if you don't, and I will explain it.
Right. Is it because, say, stressor A reduces by 5, then B reduces it by 10, or reduces by 15, so together then reduced by 20?
Correct. That is it. So this stressor reduces the richness by 5. This stressor would reduce the richness by 15. And the two together, because we're adding the impact, would reduce it then by 20, leaving just 10. Okay, everybody see that? That's what I mean by additive. Now, if it's synergistic, it makes things worse. So they've just made things worse in that diagram. If it's antagonistic, it makes things, I don't want to use the word better. It makes it less severe than, say, this stressor alone. So you're getting just a reduction to 20. So please don't go away thinking, well, antagonistic is actually good. It's less severe than synergistic, but it still represents impact on the community. Okay, so this is demonstrating negative change. Here we're dealing with, should I do this one? We're dealing with positive responses. Now, when I use the term positive, some people are inclined to think, well, positive means good. But positive in this context is only representing the direction of change. Please note that it's just the direction of change. So if you wanted to try and put something to this, you could visualize this as the number of, say, chironomid larvae, which tend to be pollution tolerant. So it could be representing changes in the number of pollution tolerant fauna. So, and the control environment is just 5. You add one stressor, because conditions might improve for them, you get 10. The other stressor might give 15, and the two combined, you end up with, say, 20 of these pollution tolerant taxa. If it's synergistic, you'll even get more of them. And if it's antagonistic, you'll get a little bit less. but it'll still be impacted relative to the control. All right. That, Louise, you're looking puzzled.
No, it's fine. I have it. Thank you.
You have it. Good, good, good. You see, if I had you in class, I'd know whether each of you were puzzled or not puzzled by it. Yeah. So that's what it means. And then this one I'm going to leave you to think about for yourselves. I'll explain what's happened here. This is showing that. So again, we have the control. And for this first stressor, the effect is in a positive direction. So you get more of whatever it is. Whereas this stressor causes a decline in numbers, and then you add them together and you get this 15. So this would be sort of +5 minus 15, and then you take it from your total. And that's how the additive works. And of course, it's always wonderful to show it on a diagram like that, but in reality, when you're working with it, it's a lot more complex. Okay, where did I go from there? All right. Okay, so just a little bit on the types of studies and why we might actually want to study multi-stressor impacts. The main reason is to try and identify the stressor that's having the strongest impact of the dominant stressor so that we can target measures to address them. And sometimes, if we're lucky, you know, the measure might actually knock out 2 stressors. But can be challenging because it depends on what biological receptors or indicators you use, because sometimes they differ in their sensitivities to and therefore response to various stressors. So in a lot of these studies, you will see that they have examined, you know, not only invertebrates, but maybe looked at algae or fish or macrophytes. So quite a variety of studies. And those studies have been carried out using different approaches. Some have been field observations, and you know, field observations are obviously much more realistic because you're seeing everything that's interacting there. But because you have so many interacting factors, it's difficult to pull out what is causing the impact, and even more important, it's very difficult to replicate. So a lot of studies are based on mesocosms or slightly larger experimental systems called artificial streams. Oops. We have worked with mesocosms, and I'll just show you those in a moment. And the good thing about the mesocosm and the artificial streams is that you can control what goes into them. So you have very well, can have very well replicated and controlled experiments. But, you know, some would argue, well, perhaps they're not entirely realistic. So I suppose the best approach is combining both mesocosm or experiments and field observations to draw the conclusions. Now, just want to go on to, now on the file that I uploaded to Brightspace, you'll see a video there showing the experimental system that we worked with. Now, unfortunately, it just doesn't work on my laptop, but you can go back. I can get the music, but I can't get the images out of it. But this is what it looked like here. So we have these dishes which are donut shaped. These are the mesocosms. And what happens is that water comes in this little jet here, and it's coming in under a force. So it spins around the mesocosm and exits down through the center of the dish. So it's simulating a little mini river channel. Okay. The water is delivered from these, from these header tanks here. And the water in this experiment that we ran was drawn from a nearby river. So we have a whole series of those header tanks. They feed the water down into these mesocosms. The water leaves via the bottom and it's channeled away from the stream and eventually returns to the stream. The other thing we have coming in this case here, you have a nutrient dripper. So behind this scaffolding, there were containers and pumps delivering nutrients to each of these channels there. Nutrient dripper, and there's one there with sediment in it. Now, there's two ways that the invertebrates we were dealing with in this case can react. They can either get out, in other words, drift out, or they can stay and die. So we captured what is called the drift. So what you're looking at here is underneath the mesocosms, and what we have suspended are ladies' pop socks. So they're attached, and that's what the attachment there. They're attached to the center, and they hang underneath these dishes. So the water exits through these pop socks, and we can catch then what is leaving. And we were catching the drift every initially 24 hours, and then every 48 hours after that. So it meant just taking off those pop socks and putting on a new pair of pop socks to each of the mesocosms. This study was carried out by one of my PhD students, a male PhD student who had to take a trip to Penny's every week to buy more pop socks. So a little bit embarrassing for him. Now, so apart from leaving, the other thing we did at the end of the experiment, we looked at what was left in the dish. So some animals would stay because they found favorable conditions, and others would have died off. Okay, so obviously it is important that you have a look at the paper. I think I put those papers up. If I haven't done so, please tell me. But I'll just give you the highlights of some of the experiment. So the first thing is in terms of the drift. The results showed that sediment was the dominant stressor, and it affected 11 of the 14 drift variables. So going back here, these are the drift variables that we looked at, you know, the total amount drifting, the types of animals drifting, the whole composition of the community drifting, the number of batids, heptagenids, and so on. So sediment affected most of those variables. And there really was no measurable effect of nitrogen or phosphorus on the drift. Then in terms of what I call the benthos, that's the animals remaining at the end of the experiment. So we had a period of treatment, so I think it was 2 weeks after which we terminated the experiment, and we had a look at what was left in the dishes. And here we found that certainly the sediment did decrease total and EPT abundance at the high sediment levels, but the extent of this decrease differed across the different sediment levels because there was an interaction with phosphorus, and that interaction was actually antagonistic. And what happened is that what we think is that the sediment was able to bind some of the phosphorus and therefore reducing the effect of phosphorus. Okay. Now we did another experiment, and here we asked, well, you know, which is most impacting, chronic pollution or acute pollution? Acute pollution would come in say rainfall events where you just get wash off of any contaminants from the surface of fields. Whereas acute, it would be, that's acute, whereas chronic would be this low level continuous input of contaminants, you know, like we looked before, the runoff from septic tanks. So every day you're getting that low level of input. So in this experiment, we created chronic nutrient enrichment by using the nutrient levels in the previous experiment and just continuously delivering those nutrient levels. And then on 2 other days, we delivered a higher level of nitrogen and phosphorus. So we sort of doubled the concentration of the chronic levels. And then we also had sediment addition at the start of the experiment. So you ended up with these 14 different treatments. Again, you can have a look at them in your own time. And we replicated each of those. So quite a well-replicated experiment. We had 26 days of what we call colonization. This is where, again, water was being drawn from the river into the header tanks down into the mesocosms, and that water always brought some invertebrates into the channel. So it allowed some natural colonization, but we supplemented that by adding a sorber, part of a sorber sample to each of the channels as well. And then we had 18 days of manipulation or experiment. So key results, again, really complex because we have so many opportunities for interaction. So in the first, so I'm going to give you the key messages that I took from the paper. And this is always a challenge for everybody to get the key messages from the paper. So in the first 48 hours, sediment was the dominant driver of the drift response. And what that told me is that, you know, sediment deposition can have that immediate detrimental effect, affecting communities in a very short period of time compared to other sources of pollution, if you have a high level of it. Whereas nutrients take a time to evoke a response, generally through growth of algae. 48 hours after the first nutrient pulse, sediment was still the main impacting stressor. But 48 hours after the second acute or nutrient pulse, there was no longer any response to sediment, but the variables were now affected by nitrogen. So nitrogen now became the main driver of the drift response. We don't know why nitrogen became the driver. It's mainly nitrate that we added. So we're exploring that a little bit further now. Now, the effect of phosphorus increased as the experiment went on. The main reason is that it takes time for the algae to grow. And I suppose overall, we concluded, you know, that certainly acute impulses have an effect, but in general, chronic nutrient pollution appears to have a greater negative effect than in acute inputs of nutrients. But we need more work in that area. Okay, so a little bit on sediment that we have covered there. And just highlighting that we can't ignore sediment any longer, because it's probably going to be a dominant stressor and it's going to induce interactions with other stressors. We certainly know that climate change is going to bring additional stressors, and there's limited research on the effects of flow and temperature changes on the multi-stressor environment. I have a PhD student who's doing work on that at the moment in collaboration with the University of Otago in New Zealand. Again, experimental work. The responses we're clearly seeing are complex, and they're going to complicate any efforts to mitigate the impacts on aquatic ecology. Oh, sugar. That one has still audio on it. What they're thinking is that climate change is likely to bring in other stressors, and it's also going to impact the delivery of stressors. So increase in rainfall events is going to bring in more nutrients in a shorter period of time. So how do we use the information we have then to move forward? And how would you advise somebody to manage stressors in a multi-stressor environment? And we're dealing with that. There's no doubt about that. So the first step, and this comes from a project known as the Mars Project, nothing to do with Mars, got to do with managing multiple stressors and based mainly on invertebrates. So we have to identify the range of stressors that are relevant to a particular catchment. That means looking at the land use, looking at whatever chemistry results we have. Then to carefully select response variables and metrics. So are we going to look at invertebrates, or are we going to look at algae? Perhaps we need to look at both if we're going to get a good measure of what is being impacted, because an impact on algae can feed through to an impact on invertebrates. Then identify the minimum number of stressors that need to be tackled. identify if it's possible and prioritize the dominant stressor. So you're looking for the stressor that accounts for most of the effect. So that's the one you would try and mitigate first of all. But if your stressors are showing similar effect sizes, then you need to give priority to both. If stressors interact, then you have to identify the type of interaction. And the difficult one is antagonism, because if you remove one, you might for a period of time have things a little bit worse. So what they're saying is that identify the stronger stressor or the non-antagonist. And antagonists are stressors that dampen the effect of other co-acting stressors. For example, high flow dampens the effect of nutrient enrichment. So reducing the antagonist without prior mitigation of other stressors like non-antagonists would result in an aggravated stressor effect. And then finally, you have to think about how the stressors are delivered. Okay, and that schematic then really summarizes what we've just said. Looking at the range of stressors, prioritizing them, looking at whether there's a dominant one, types of interactions, and then dealing with the outcome of that. Okay, so I'm going to leave it at that for today because I have to join an advisory session for our first years in about 10 minutes. Does anybody have any questions? What I will try and do is, if you go back to some of the previous slides that I haven't gone through, because they're just summarizing some of the key findings from previous papers, some of these here. I'll try and get those papers onto Blackboard or onto Brightspace for you, because those are the key studies that have been carried out to date on the effects of multiple stressors. In particular, there is a recent one by this guy, Burke et al. That's a 2020 paper, and that really brings together a lot of information on multiple stressors. And in particular, the differences between rivers and lakes, really pointing out that for rivers, the effect of nutrient enrichment really is causing a lot of the biological response. Any questions?
Yeah.
Yeah.
I'll go really quickly. The bioassessment report, you had a document up on Zoom the last day. Are you going to be uploading that?
I am indeed, yeah.
Okay, brilliant.
All that in my diary to go in tomorrow. Yeah.
Okay, brilliant. And is that again in the same groups? Are we supposed to do one, or
yes,
we each
please
do one?
stay in the same groups, yeah.
So each group uploads one draft and then one final.
One report, but every individual. So you're going to upload a draft initially, and I will give you a response to that, and then you amend it. After that, each individual writes a single abstract. So the abstract is for the individuals.
Okay. Great.
Okay. So I'll upload that tomorrow. It's just my, the reason I'm here today is the guys are looking at my internet connection. So hopefully that's going to be a little bit. Could we leave on Friday briefly at 10 o'clock, because I want to go through the monitoring program exercise with you. So 10 o'clock, and then that I think brings us more or less to the end of what I need to cover with you. But obviously, when I've gone through the draft reports, I'll meet you again and go through the main issues with the draft reports. Or if you have any questions on anything else. And clearly, as soon as I know the final arrangement for the exams, I'll let you know. But either way, there will be essays on that paper, and those essays are given to you in advance of the exam. I've always done that. So it's not about so much about memory work as preparing and researching.
Mary, can I just ask a question? You know the policy brief that's due on Sunday?
Yes.
Is that just pretty much us, like a page long of like us looking up like stuff and giving like bullet points like this is what we should do to like try and, or this is like the reason to protect it and all that? Sorry.
Yeah. So you imagine your local politician, And he asked you, well, why should we protect biodiversity? That's what you're delivering to them. Something that is well supported by science and is understandable. So it's converted into non-technical language as far as possible, but that it's well supported.
They're pretty much trying to use like simple language as though like someone who's never done anything like this before can at least like.
Yes.
I get that, yeah, kind of like that kind of thing.
Yeah, but don't dumb it down. It's make it understandable. It's the main thing. Okay.
Okay, yeah, I just wanted to make sure.
Is it just biodiversity, sorry, or could you be for drinking water, domestic uses?
No, I give you biodiversity as far as I remember, isn't it?
Okay.
Yeah, no, not drinking water. But you can always make the link to good biodiversity and water quality. Remember, biodiversity underpins the processes that secure good water quality. Yeah, so making that link is good. And really, if you can make the link to benefits to people, that's much more convincing, and that's the challenge. So ecosystem services and benefits.
And about the referencing, do you want points referenced, or should we just have a list of references at the end?
I would say if you maybe put the little subscript, and then down at the bottom of it, put in the references in a smaller font.
Okay, so number is referencing.
But keep the number. I think you're given 5 reasons to protect freshwater biodiversity, so maybe keep the number down to just the key references. So if somebody was going to look it up, I don't want to look up 20 references, but I just want to get me to the key references.
Okay, so if it's something kind of straightforward and understood as fact, you don't have to put a reference. Or something kind of generally known.
Yeah. But have some supporting references in there.
Yeah, okay, thanks.
All right. Okay, if you have any other questions then on Friday for that, we can address them. All right. I'll leave you and go and talk to my first years.
Thank you.
Thank you.
Thank you.
Thank you.